Welcome to this lesson on the anticodon, which is the tRNA recognition site. The anticodon plays a crucial role in protein synthesis by ensuring that the correct amino acids are delivered during translation. Understanding how these three bases work will help you grasp the fundamental mechanism of how genetic information is converted into functional proteins. An anticodon is a sequence of 3 nucleotides found on transfer RNA, or tRNA molecules. These 3 nucleotides are complementary to a codon on messenger RNA, or mRNA. The anticodon acts like a molecular key that fits into a specific lock, which is the codon on the mRNA strand. This complementary pairing ensures that the right amino acid is added to the growing protein chain. The anticodon is located at the bottom of the tRNA molecule, opposite to where the amino acid attaches. Think of the tRNA as having 2 important ends. The top end carries the amino acid that will be added to the protein, while the bottom end has the anticodon that reads the genetic code. This structure allows the tRNA to act as an adapter molecule, connecting the genetic information in mRNA to the actual building blocks of proteins. The anticodon consists of exactly 3 bases, just like codons on mRNA. Each position in the anticodon can contain one of 4 nitrogenous bases: adenine, uracil, guanine, or cytosine. These are abbreviated as A, U, G, and C. The specific combination of these 3 bases determines which codon the tRNA will recognize, and therefore which amino acid will be delivered to the ribosome during protein synthesis. The anticodon recognizes its matching codon through complementary base pairing rules. Adenine always pairs with uracil, and uracil pairs with adenine. Guanine always pairs with cytosine, and cytosine pairs with guanine. These pairing rules are based on the chemical structure of the bases and the hydrogen bonds they can form. This specific pairing ensures accuracy in translating the genetic code into the correct amino acid sequence. Let's look at a specific example of how codon-anticodon matching works. If the mRNA has a codon with the sequence AUG, the corresponding tRNA anticodon will have the sequence UAC. Notice how each base pairs according to the complementary rules. A pairs with U pairs with A, and G pairs with C. This precise matching ensures that the tRNA carrying the correct amino acid for that specific codon binds to the mRNA. An important feature of codon-anticodon pairing is that they run in opposite directions, which we call antiparallel. The mRNA codon is read in the 5' to 3' direction, while the tRNA anticodon runs in the 3' to 5' direction. This antiparallel arrangement is similar to how the 2 strands of DNA run in opposite directions. This orientation is essential for the proper binding and recognition between the codon and anticodon during translation. To summarize, the anticodon's 3 bases are fundamental to accurate protein synthesis. By matching precisely with the mRNA codon through complementary base pairing, each tRNA molecule delivers the correct amino acid to the growing protein chain. This molecular recognition system ensures that the genetic code is translated accurately, allowing cells to produce the exact proteins needed for life. Understanding this mechanism is essential for comprehending how genetic information flows from DNA to RNA to protein.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.